Good evening everyone. Now I am going to explain about the Corynebacterial diphtheria. What do you mean by Corynebacterial diphtheria? Corynebacterial diphtheria, it is a club-shaped bacteria which causes the disease called as the diphtheria in the humans. So in this chapter what we are going to explain about the introduction, morphology, cultural characteristic, pathogenesis, laboratory diagnosis, prophylaxis and immunization and other treatment. So now come to introduction part. Corny means club-shaped. It is a gram-positive bacteria. It is a non-sporing, non-capsulated, non-motile. So different species of this Corynebacteria are called as Corynebacteria diphtheria, Corynebacteria ulcerans, Corynebacteria minutissimum, Corynebacteria tetanus, and Corynebacteria pseudodiphtherians. And now how we will remember this. It is remembered as one code, that is dip ulcer minute 10 pseudodip. Okay, dip means Corynebacteria diphtheria, Corynebacteria ulceranus, and minatissimus, tenus, and pseudodiphtherism. And after that come to morphology. How the Corynebacteria will looks like and how it will be. So it will be similar like bacillus. Bacillus means rod shape. So it is also rod shape. It will be measuring about 3.6 to 0.6 to 0.8 mu meters. So granules, so each and every end it will be like rod shaped, and each the bacilla will be appears in the form of the pairs, and at the end of the each pair there are the granules are present. These granules are called as the polar bodies, and these granules are composed of polymetaphosphate, and hence it is called as metachromatic, or it is called as metachromatic granules. Why it is called as metachromatic granules? Because when we are adding methylene blue, the granules will take the blue stain, so it is called as metachromatic or enriched granules. Now we will why it will be appears in the pair, because due to the incomplete separation of the daughter cells after the binary fission. Now come to cultural characteristic. What is the cultural characteristic? It is the aerobic, which will grow in the presence of the oxygen and facilitative anaerobes, and it will be grows at the temperature of about 37 degrees, ranges about 15 to 40 degrees, pH at 7.2, optimal temperature, optimal pH. So on the different media, how is the growth of this Corynebacteria diphtheria? So on the nutrient medium, we come to nutrient medium. So in nutrient medium, blood, serum or the sugar have been added, so the growth will be scanty. So his serum water, I have mentioned that his for him, that is the code for fermentation of pneumococcus. So his serum water test, this is liquid medium. So in this one Corynebacteria diphtheria, it will show the turbidity and the pellicle formation it will show. After that Lofer's serum slope. In this one how the diphtheria will grow, it will grow very rapidly. Colonies appears after the 6 to 8 hours of the incubation, long before other bacteria will grow. So the colonies will, how it will be small, circular and white or the grayish. Now see the selective media. In selective media, the selective media tellurate blood agar. So what the selective media it will do? It will actually, we know that the selective media it will inhibits the growth of other bacteria. So it will grow very slowly and form grey to black. What is the reason behind this? Due to the reduction of all the phosphate tellurate. There are the three different types of diphtherians. What are the three different types of diphtheria? There are gravis, intermediates and the metis. Metis means 1 to 3 strains are called as metis. Intermediates means 4 to 6. After that gravis is called. So now come to tensile agar. It contains the sheep blood, bovine serum, cystine and the potassium tellurate. How the growth of the diphtheria? It will grows as a black or brown colonies are formed. So on the brown colonies, it will be useful for differentiating all the different types of diphtherians. So now black, it will be a black colony with halo. Without halos, staphylococcus will be formed for the black colonies. Now come to toxins. What are the different types of the toxins are produced by this? So the toxins can be produced by the Corynebacteria diphtherians are very powerful, that is the exotoxin. So 90 to 95% of the gravies along with the intermediates are the toxic genus, and 80 to 85% of the meter strains are also toxic. Now the toxicity, how it will be, on which it will depends, how it will forms. So there has one of the code, it is called as ABCD. So atypical, E. coli, botulism, toxin, cholera, diphtheria, and varroa cytotoxic. So the phase it will be depends upon the phase coded. Phase coded means beta phase carrying toxin, and it will be depends upon the, and we know the two different type of fragments are present on the toxin A and B. In this one B, it will be attached to host. The toxin has the fragment A and fragment B, and B is attached to the host cell, and it will attaches and it will be penetrate into the host cell, and after entering into the host cell, what it will do, it will prevent the protein synthesis by the ribosome formation. It causes the cell death in the cell membrane. After that, we come to again where it will depends upon the iron concentration. If there is the concentration of the iron is more, it will inhibit the toxin synthesis and regulating the diphtheria toxin. It is called as repressor. So gene in the bacterial chromosome are also, it will depends upon the gene also. So the toxide, some of the toxide are useful for the vaccination. So this toxin is antigenic and antitoxin, so it is called as protective. Virulent cannot be given directly by the vaccination because toxin have to convert into toxide. Toxide form means should be in toxin form, so that virulence, whatever it will causes the diseases, it will get loss. It can be promoted by the toxin be promoted by formalin, acidic pH, and prolonged storage. Now Park William Yates strain, it is one of the source of toxin for preparation of vaccine. It is one of the source. So now come to LF unit. LF unit means it is a loafers fluctuating unit. It is the amount of the toxin. Now come to pathogenesis. We are discussing about the pathogens. What are the as we know that pathogenesis means Corynebacterium diphtheria, it causes the diseases. After that pathogenesis, in this